Good morning, all. Today is Monday, June 15th. Hopefully you had a good, safe, and healthy, fun weekend. We have a busy week ahead of us, so hopefully you're well rested for it. Today we are going to finish up the rest of our cells with our last major physiological process, and that is going to be protein synthesis. As we will see and as we'll talk about, this is a majorly important physiological process, so something that is almost certainly going to be on the exam. And one of the important things to remember is that when we have, especially when we have two very similar related processes, that always relies itself being the smart, sophisticated students you guys are going to become, that pretty much lends itself to three possible essay questions, right? You're going to need to describe process A, describe process B, and compare and contrast processes A and B. So always be thinking about that when you're working on these types of things. Our second goal for today is going to be to get our practice lab exam under our belts. So that'll be an opportunity for you guys to take that practice lab exam today. Again, you're not going to be video recorded during this one, so I've made it a little simple that way, but we're still going to lock down the computers and have some test type of conditions for that. But again, there is no points on this. This is just an exercise for you to give you a chance to gauge how you are preparing for this class, but also for you to see what to expect from the lab exam so that you'll have a chance to see that. Like I said, all of these are questions that have been on previous lab exams. Some of these questions could easily be in the text bank that you're going to get. And again, remember, that's one of the important things about this exam. That one of the things I don't like about this online format is I don't have as much control over the exams, right? Obviously, when I write the exam, I know exactly what's going to be on the test, and I can set it up the way that I want to, precisely to the word, to the letter, to the exclamation point, to the period, to everything. Here, every single person is getting a completely different test with random questions assigned to them. And so just inherently, there's a massive test bank of questions that it is going to be selecting from. And so unfortunately, one of the challenges of that is that not every test is necessarily going to be equal in its and how hard it is. Some may have a few more easy questions, some may have a few more hard questions. The good news about the randomness of this is that by the end of the semester, everybody should have about the same amount of easy and tough questions. So it'll all work out at the end, but each individual test is a much more variable. So that is more challenging for you as a student because you really have to be prepared for everything. So this will give you an idea of some of the questions that are in the test bank, how I've asked questions in the past, how I'm going to ask questions on you, because on the real exam, because as we mentioned, the goal is for you to show me you have the information. The goal is not for me to be tricky or challenging because I don't have to do that. The class is hard. So this is a way of showing you the process. So hopefully the process doesn't get in the way of you showing me you have the information. So you will take that practice exam after lecture today, and then we will then meet back after that. We'll take both our break and the exam and then the practice exam, and then we'll come back and talk about that practice exam. And then from there, hopefully we'll have enough time left where we can then, the third thing, our goal for today is to start our introduction into tissues, just to dip our toe into it to get us a head start for this week, because we got a lot of material to cover this week. You also have a fair amount of assignments, not quite as much as you had last week. There was a lot of getting started assignments as well as other assignments that we had, but this week we still have some things to keep you busy. The first Tuesday is your PhysioX exercise 1 is due. Remember, there are 5 activities. That means 5 lab reports that you are submitting on Canvas. The other thing that I want to strongly encourage you to do before class on Tuesday is to print out your epithelial tissue and connective tissue handouts. This is where you're going to be drawing your tissues and labeling your tissues. This is 2 things. It is an assignment that you are going to be taking a picture of and submitting to me. It is due at the beginning of class on Friday. But the other thing this is your study guide for the tissues portion of our lab exams. So it is important that you have this as an important study tool. And while I am not going to take the time in class for us to completely go through and have everybody do perfect drawings and everybody doing perfect labeling, it is important that we do go through it together so you understand what it is that I'm looking for and how to fill it out so that you can be successful. So having those printouts so that we can work with them together, so you can be looking at them while we look at the histology slide and we work on that together, is something that is going to be very important. And it's also an assignment that is due on Friday. On Thursday, your unit review 5 is due, as is a biointeractive on skin color. So for that skin color, what you're going to do is you go to follow the directions on the link in module. Remember, the link is in the modules. There's a handout there. You're going to print out the handout. You're going to complete the handout as you watch the video, fill out the handout, and then submit that handout to me. And then on Friday, your unit review 6 is due. The epithelial tissue and connective tissue handout is due, again, with drawings, with labels. You're not required to use color, but color is, again, something that can be useful for you, but you are required to have labels. And again, it's like the unit reviews that you guys have been turning in. I want written labels. I don't want numbers and then numbers up for the things, or lines or colors or anything like that. That's not how you learn. That's not how you use this as a study tool. You need to write out the labels. When you have an image, you need to label that image. So make sure you do that for full credit. Also, we are going to do, obviously, we're not meeting in the classroom. You can't do the exact fingerprinting activity that you have in your lab manual. But what you can do at home is finding a pencil or finding a pen or something like that. Hopefully you have an ink pad, but if not, there are other ways you can color your finger. You are going to be making fingerprints and using the chart that is on page 146 of your lab manual. I want you to identify the basic pattern of your fingerprint, and I want you to find, I don't remember how many it is, 2, 3, 4, 5, something like that. I think it's 3 or 4 of the minutia of details of those. So I want you to be circling them and labeling them. You can do yours, you can do someone else in the family, do it with your family. It's a fun activity. So that is going to be due. So I want your labeled fingerprints due on Friday as well. And those instructions, like I said, are in your modules. And also, like I said, you're going to be using page 146 of your lab manual for that. All leading up to Monday, when we're going to have the lab and lecture exam. Again, we're going to be using Proctorio. This is going to be the exact same format that was used for the chemistry quiz, that was used for the Proctorio and syllabus quiz. So you are going to be video recorded. You are going to be audio recorded. Again, it is vitally important that you scan your entire workspace. I want to see the countertop or the desktop, where you have what's behind the screen, right? I don't care what's in the corners of your room. I don't care what color your curtains are or what books you have on the shelves. That's not what I'm interested in doing. So don't hold it up and spin it around like a lot of you have been doing. I want to see your work area. So scan downward so that you can show me that all of your materials are put away. You don't have anything out. You have a clear space so that the information that is being provided in the exam is yours and not coming from another source. And again, the other thing, remember, we are not meeting on Monday. Monday, you will not be seen, and hopefully you won't be hearing from me at all. You will be taking the exam and doing the exam. It needs to be done between 8 and 12:35. On the off chance that you have problems with the exam, there is a way to contact Proctorio while you're taking the exam. They are the people that you want to contact. If you get, for some reason, you get kicked out of the exam, they're the ones that can get you back into the exam. I do not have the means of doing that. So if you have some issues, talk to Proctorio first. Use their help. They're very good, very fast at responding. And then if there's some other issue, then you want to reach out to me. Right? And of course, don't be texting me or emailing me on your phone while you're taking the exam either, right? Get through the exam, or if you get kicked out or something, that's when you contact me. But the Proctorio is very good at helping people. So use their help to get into that. And so, like I said, hopefully you won't hear from me. I won't hear from you on Monday. And then we'll meet back on Tuesday and dive into the skeletal system.
Quick question.
Yes.
When we come back on Monday and we're about to take the exam, is the exam going to be in the module or?
No, so the same, you'll click on the quizzes. So on the quizzes, you click on the quizzes tab, and that will get you to where the quiz is and where you can complete it there.
Oh, gotcha. Okay.
Yep. All right. Any other questions on that? All right, excellent then. Then that's our game plan. Let's get started.
Oh, actually, I'm sorry. I do have one question.
Yeah.
Besides everything that you typed up right now, are we getting any other kind of like reviews, study guides or anything like that, or this is it, correct?
This is it. So like I said,
Okay.
as I've said, your lectures, you are responsible for everything we talk about in lecture, for all the lab activities that you're responsible for. So
Okay.
again, if I were to write all that out, I'd just be recreating the lecture. So
Gotcha.
the good general rule of thumb to remember is you are responsible for everything.
Okay.
All right. So great question. No. So again, think of this in terms, when you're taking the exam, think of it in terms as if you were taking it in the classroom, right? Like I said, I know you are at home. And so, but if we were in the classroom in the lab, you wouldn't be able to eat, you wouldn't be able to drink during the exam. So I encourage you to not eat. And I'd prefer you not have a drink nearby. I'd prefer you not smoke or vape or anything like that during the exam. So again, you won't get a 0 if you take a sip of coffee while you're taking the exam, but I do want to strongly encourage you not to do that. However, what you can't do is get up and leave during the exam. Because again, you know, whatever reason you may need to, if going to the bathroom or something along those lines, you have to appreciate that I don't know what you're doing once you're on camera, and I don't want you taking your camera with you into the bathroom. So that's a no-win situation for everybody. So you have 2 exams. You are welcome to take a break in between the 2 exams. So go to the bathroom before, take one of the exams, go to the bathroom after if you need to, and you'll have it set up that way. As far as the time goes, great question. You have to complete them between 8 and 12:30. The lab exam, I think, I don't remember. I'm not trying to be coy. If I knew the number of questions, if I remembered the number of questions, I would tell you. I think it's around 70 questions. So I think my guess is you have somewhere around 80 minutes to be able to take that. So let's just call it an hour and a half because that's a simple number. It's going to be an hour and a half or less to take the lab exam. You have 2.5 hours to take the lecture exam. So that right there is, if you put that all together, that's 4 hours. Now, nobody should take 2.5 hours for the lecture exam. That 2.5 hours is partially in place. If you have problems, you get kicked out, things along those lines. But really, with the number of questions and stuff that's there, it shouldn't take you that long. If you're a really slow typer, or if you have technical issues, it takes a little bit longer. So that's why I've given you more of a buffer. Even the lab exam shouldn't take you that long as well. But if you think about it, you have 4 hours of exams and 4 hours and 35 minutes of the class. So if you start close to 8, you know, 5-10 minutes after 8 o'clock, and then you take a good 20-minute break in between, you can still easily finish with plenty of time for that to be able to complete everything within the class time. All right.
Quick question.
Yeah, wait, hold on. I would encourage you to start earlier rather than later, just in case there are technical issues, because the time cannot be extended. So if you get kicked out and it takes you an hour to get back inside, that hour counts as part of your time. And so the exam cannot be extended. The proctorio is a necessary evil we need to do to maintain the integrity of this class, but one of the problems with it is it doesn't have a lot of flexibility. So it's not I can't extend the exam 30 minutes for you because you had problems with it. It's just that the mechanism isn't there in place to allow me to do things like that. So I don't have as much flexibility. So I do encourage you to start sooner rather than later in case you have any issues. All right. Yes, you had another question?
Yeah, you mentioned that the lab is 70 questions. Is it
Don't
like
quote me on that. I may be off. I don't remember exactly.
right?
It's around there. Yes, it's around there.
And are they gonna be all multiple choice or fill-in as well?
No, the lab exam is all fill in the blank, and spelling counts, as I mentioned. So you are
Right.
going to be writing out the answer. This is not multiple choice. A lecture exam will have multiple choice. We'll have some fill in the blank, and we'll have essays. But all the lab exam questions, as you will see when you take the practice exam, it's just they're called essay questions, but really most of them it'll be 1, 2, 3 words that you'll be writing to answer the question.
Okay.
All right. Questions on that? And last thing, remember, like we talked about, be specific. Well, we'll talk about it when we get to the quiz. All right. Any other questions? All right. Let's dive in to our last major cell physiological process. And as I said, that is going to be our process of protein synthesis. Now, if you've taken some kind of biology class, again, what they talk about is this rule that we have those 46 chromosomes that we talked about, and each one of those chromosomes have thousands of genes on them. And those genes are sequences of nucleic acids that provide information for producing, and again, the general rule you hear is one gene produces one protein. However, being the A&P students that we are, we're a little more sophisticated than that, and we understand that a fully functional protein is typically made up of subunits. So really what one gene produces is one polypeptide, one single sequence of amino acids, which we know is going to be cut, is going to be folded, and becomes a unit that is used to make our fully functional proteins. As we talked about, this process of protein synthesis requires two steps. Again, what you have to remember is that we have this issue. We have a cell, and inside that cell, there is the nucleus, and that those genes are contained here, right? The DNA is contained here inside of the nucleus. The problem is, we know out here is where the rough endoplasmic reticulum is. And as we know, that rough endoplasmic reticulum is rough because it has those ribosomes on it. And that is where we are going to make the proteins. So we have this issue where the information that we need is locked up here inside of the castle. And yet out here are the workers that need to do the work. It's not that different from the way things were in medieval times. In medieval times, right, information was power. Information was vitally important. So books would be locked up in places like churches, like castles. And if you wanted a particular piece of information, you would go to that castle, you would go to that church, and you would plead with the people and present your situation, your case. And if they deemed you worthy, they would provide you with the information. And when they provided you with the information, did they give you the entire book and, like, you know, you checked it out with a library card and you got to leave and bring it back 2 weeks later? No, typically what they would do is just provide you with the bit of information that you wanted. And they wouldn't do that by ripping it out of the book. They would make a copy of that piece of information, a portable, temporary copy of the information, which you could then take with you. They had special names for those people. What did they call those people who made the copies of the information, the copies of the books?
Scribes?
Right.
Scribes, absolutely. And one of the amazing things is that most scribes were illiterate. They couldn't read. Because again, knowledge was power. They didn't want that knowledge getting out. All a scribe did was see a word and copy it on a separate piece of paper exactly as they saw it. They had no idea what those letters meant. They had no idea what the words were. Their job was just merely to copy that information, right? That was their job. They were the scribes. And in fact, that is going to be the first step in our process. The first step in our process is transcription. To take that information from that permanent copy, that locked up copy, the DNA, and convert it into a movable temporary copy, RNA, and specifically what we call messenger RNA. RNA, which once you write out once, you may abbreviate mRNA. That messenger RNA would then leave to be able to go to the ribosomes, and once they get to the ribosomes can then turn it into the proteins that we want. Now, if you were to go back in time to one of those castles, and when you got to that castle, you were able to then ask them for the information, and they deemed you worthy, and they made the copy of the piece of information and then provided it to you, would it actually do you right now any good?
Not really.
Why
Because you
not?
can't translate it.
I'm sorry?
You can't really translate it.
Well, exactly, because most of the time, again, back in medieval times, most of the learned scholars, most of the information that was stored was written in one of two languages, right? Greek, maybe if you were lucky, but most of it was in Latin. And how many people here speak Latin?
Nobody.
Nobody, exactly. Nobody speaks Latin. So
We all
once,
speak,
I'm
right?
sorry?
So we
Someone
all
speak
kind of speak that, right? Homeo, hypo, hyper.
There you go. So we can know a little of it. Yeah, absolutely. We do some roots and stuff like that. Excellent, right? But for most of us, that information would then need to be translated, where we change the language. And that's really the key. Here, we are going to change the language. Notice before, we didn't change the language. We went from DNA to RNA, but those are both nucleic acids. This is still nucleic acid to nucleic acid, a permanent to a temporary, but we didn't change the language. We just copied the information. Here, the second step in this process is translation, where we change the language. We change it from the language of nucleic acids, the language of DNA, to amino acids, the language of protein. And like I said, the transcription is going to occur within the nucleus, and our translation occurs typically at the rough endoplasmic reticulum, and more specifically with those ribosomes. And again, if you don't like my pretty picture, here's the pretty picture from the textbook that shows that same process. Step 1 is transcription that takes place here in the nucleus. Step 2 is translation that takes place at the ribosome. And remember, most of the ribosomes are located in the rough endoplasmic reticulum. Of course, the key word there is most. Again, most means not all. So remember, we did talk about there are some free-floating schools of fish type of ribosomes, although they don't clump in those schools of fish, but there are free-floating ribosomes inside the cell. But most ribosomes are in the rough endoplasmic reticulum. All righty, let's talk about these two processes then, and let's talk first talking about transcription. Again, transcription is where we are making a copy of the genetic information. Again, we are going from the permanent to the temporary in this, but we are using the same language, nucleic acids. So it is nucleotide to nucleotide. And the good news is we already know a little bit about how this process would work. We know a little bit about our nucleic acids. We know that with DNA, how many different nucleic acids are there again that are used in DNA? 4, exactly. And again, just let's go to the letters. What were the letters again?
A,
T.
T,
A, E, E.
There you go. I heard them all. A, C, T, and G. And actually, let's cheat and change the order. G and T. Excellent. Right, those are the 4 nucleic acids that are used, or nucleotides that are used to make DNA. And as we also talked about, RNA also has 4. They also use A, C, and G, but instead of thymine in our RNA, what was the nucleotide we used instead?
Uracil.
Excellent. Excellent. So we know that there are going to be some subtle changes in that we are going to see. But we still understand the basic base pair rules. We're actually going to use processes that are actually very similar to what we saw in DNA replication, where it's going to require dozens of enzymes that are going to be involved in this process. But just like in replication of the DNA, the primary enzyme was an enzyme called DNA polymerase. Well, here in transcription, we are going to use an enzyme called RNA polymerase. It is similar to DNA polymerase, where the 2 strands are separated. We make a copy of the information. The difference is in DNA replication, those 2 strands stay apart and become the templates for the 2 new strands of DNA that are made. However, what our RNA polymerase does is just temporary, and that's really the key here. It's going to temporarily pry apart the DNA, make copy of the information, and then the DNA goes back together. So in this process, unlike the previous process of DNA replication, where 1 strand becomes 2 strands, in transcription, the DNA is unchanged by the process. So that's going to be an important distinction here. But in many ways, the process is the same, or at least similar to what we saw in DNA replication. Now, again, as we know, when we talked about DNA, there are 4 nucleotides: adenine, thymine, guanine, and cytosine. 2 of them, the large ones, are purines, that is their class of bases. 2 of them are pyrimidines. They're made up of a single nitrogen ring, and those are thymine and cytosine. And we know that there are complementary base pair rules of how these come together. Adenine only binds to thymine with 2 hydrogen bonds. Guanine only binds with cytosine with 3 hydrogen bonds. So one pyrimidine binds with 1 purine, A to T, G to C. But as we also just finished mentioning, the big difference in RNA is that in RNA, what forms the hydrogen bond with A is not thymine, but actually uracil. And as you can see, uracil is very similar, not identical, but very, very similar to thymine. So thymine is used in DNA, uracil is used in RNA, but as you can see, because they're very similar in their style, it is able to form those same 2 hydrogen bonds with adenine, so it will link up. So that's its complementary base pair. The only difference with RNA is any place you would normally put a T, instead you're going to put a U instead. Okay? So when we talk about this process of transcription, basically what we are going to do is have our double strand of DNA. So let's start here. And let's just make a random strand. Oh, I should have used capitals for all that, so cheat and do that again. There we go. And we have our second. We know we are going to have, let's do this. And then we have our complementary. Oops. Nope, now I've got my caps on. So what would the complementary strand be? What goes here?
CG,
Okay, oops. G, then what?
C.
What?
G goes to C,
Get it?
right?
Yep. Then?
Is T, or T would go to A and A would go to U?
T. No, well, again, remember we're still doing DNA. So hold on, let's go back. Yeah, let's make sure our DNA. DNA strand. Okay, perfect. So this is DNA, and this is the complementary DNA strand because we know DNA is with a double helix. So what's the next letter?
A.
T,
T, remember T. Then what?
A,
A,
Yeah?
G,
G, C.
A, T, G, T, C, G, A.
Excellent.
Oh, looking at the top.
All right, so we're doing our complementary base pair rules here. Excellent. Now, one of the important things to remember is we have this double strand of DNA, but only one of the 2 strands actually has the information that we want. So in fact, what we call that is that this DNA strand would be what we call the template strand. This is the one that actually has the genetic information that we would want. It has a complementary strand because all DNA comes as a double helix, and this complementary DNA strand is what is often referred to as the coding strand. So we have our template, and the template is the one that we are interested in. That is the one that has the information that we care about. So when we peel these 2 apart, what we're going to do is we are going to separate them and then use that RNA polymerase to use our base pair rules to make a copy of it. And we're going to do that using, so let's change the color for this now. Now, I unfortunately clicked off of that, so I can't separate them anymore. So let's pretend we separate it, and we'll pretend we separate it by making the RNA above it. So again, we're going to make it above it because it's going to be convenient and equal and easier that way. But what actually happens is these get separated from each other, and we'll show a pretty picture that shows that in a minute. But if we were to do this, what would the complementary RNA nucleotide be that would match up with C?
G.
G, excellent. And what's the RNA nucleotide that matches up with G?
C.
And what's the one that matches up with T?
U.
A.
Okay, I heard both. I heard U and I heard A. Which one is it?
U.
U.
A. Remember,
A.
A is a nucleotide in RNA. Remember, in RNA, anywhere we would use a T with DNA, T with DNA, we use a U with RNA. So where A matches up with the T, what matches up with this A right here?
U.
U. And then U. And then what's the next ones?
Is that on B?
A.
And then
G.
G. C.
A.
A.
U. G.
G.
T.
Ah,
U.
U. There you go.
C.
Mm-hmm.
G.
Mm-hmm.
A.
Perfect. Excellent. All right. So there you go. Why is that not match up? Do we put one? Oh, the U's are just bigger than the T's. That's why. So it looks a little weird. All right. So again, this isn't translating as well as pretty as it would be on the board, but you guys get the idea. What we did very simply right there is exactly what's going to happen in the process of transcription. All right. Except, like everything, it's always going to be a teeny bit more complicated than this. Because remind me again, how many chromosomes do we have? How many chromosomes do we have?
I'm missing one.
46.
46 chromosomes. Excellent. We have 46 chromosomes, right? And each one of those chromosomes has thousands of genes on them. We have well more than 20,000 different genes that are found on all of our chromosomes. We have many, many thousands of genes, so much more than 20,000 genes that are found on our genetic material. But here's the problem. As impressive as those numbers sound, of all the genetic material you have in your nucleus, only about 1.5% is genes. This is an astronomical number if you think about it. That means the vast majority of the DNA that is in your nucleus is actually what used to be referred to as junk DNA. Just repetitive patterns of meaningless nucleotides that didn't seem to have any function or rationale. Now, there is a recent field of study called epigenetics, which is looking at some of the potential implications of what this is, and that is a fascinating field of study. It has nothing to do with what we're talking about today, but it is an interesting field. But the point is, if our goal is to make proteins, that's a tough roadmap to deal with. If you think about it, if I randomly threw a dart at my DNA, what are the chances that I would hit a gene, let alone the gene that I want to read? Right? If it's only 1.5%, what are the chances of me actually hitting a gene with a dart?
Very poor.
DNA? It'd be very, very small, right? So what we need is a way to help this process get started. So when we talk about the process of transcription, transcription is going to have 3 main steps with it. And those 3 main steps are going to come back to our DNA. When we look at our DNA, and let's go ahead and start this again down here. We'll start first with our template strand. That template strand of DNA, and again, remember, these, I put 10 or 12 or whatever different nucleic acids that are here for the, to make this these nucleotides together. Most genes are made up of thousands of nucleotides. So we are just looking at a small portion of them. So when we have things like the C and the G and the A and the TT and the A and the GC and then AT and GT, CGA. So I think that's the same one that we had before. That template strand, and again, let's remember we're talking about of DNA. We need a way to help the enzyme and the other proteins find the information that we want. And so what that means is there is going to be an initiation zone. That initiation zone is going to be a specific sequence of nucleic acids, of nucleotides, something like AAA, TTT, or something along those lines. There is going to be a specific sequence, and this specific sequence is going to be what our RNA polymerase is going to look for. So we need that initiation zone. All right, let's clear the picture and look at the pretty words. So again, we have that RNA polymerase that is going to unwind and copy the DNA using RNA, using, like we just did in our practice, using A's, C's, G's, and U's. Oops. C, G's, and U's for that, to make that strand of RNA. But like I said, we need that initiation region, or what we call the promoter region. That promoter region, as I mentioned, is a sequence of nucleotides. And again, some of the common ones are like AAATTT, or it could be ATATAT, something like that. There's going to be some specific sequence that tells our RNA polymerase, this is the place where you bind. So step one of transcription is to find that starting point, find that promoter region. Our RNA polymerase finds that promoter region, and once it finds that promoter region, it binds to it. And that is step one in our process of transcription. Once it binds to it, with the help of dozens of other enzymes, it is going to literally do exactly what you and I just did. It is going to pull apart the two strands of DNA, and again, only read the template strand, right? It's going to open it up, and when it opens it up, it separates the two strands, and then it makes a copy, but just one copy, and only of the template strand. Oops, stupid cap lock. So it's going to make one copy and one copy only, and that copy is just going to be of the template strand. And it's going to move base by base by base, just like we did it. This process, our second step of transcription, is what is known as elongation. We're using those same complementary base pair rules, right? We are going to, one nucleotide at a time, we are going to add A's and G's and C's and U's to the strand, making that complementary temporary copy of the genetic material. Now, what that means, and I'll use my little highlighter here, is right here is a very special region where we have this combination of both DNA. And notice they've given us these kind of color codes to remind us that not only is the difference in which nucleotide nitrogen bases they use, but remember our DNA's backbone is made up of phosphates and sugars, but the sugar is deoxyribose. Where remember our RNA, the sugar is ribose. So it still has a ribose and a sugar and phosphate backbone, but RNA uses ribose as the sugar, DNA uses deoxyribose as its sugar. So using those complementary base pair rules of A, C, G, and U, we then add new strands of that RNA, And this special region right here, where there is this mixing of DNA and RNA, this area forms what we call the DNA-RNA hybrid. Now, this is a very simplistic picture because it serves and meets my purposes, but we do have the pretty picture from your textbook, and let's look at the pretty picture from your textbook. Here we see the pretty picture of the textbook. Here in brown, we have that RNA polymerase. Notice this is where we are unwinding the DNA. This is where we're unwinding the DNA. This is where the DNA is going back together. So notice the DNA is unchanged by this process. It gets unwound. Here is the template strand, and here is that complementary strand, or remember what we also called the coding strand. And so using our base pair rules, if yellow's A, then it's adding U, uracil. If blue is G, then it's adding C, cytosine, right? Using those complementary base pair rules, it matches up the RNA nucleotide with the DNA nucleotide, right? Sugar is deoxyribose and phosphate. Sugar is ribose and phosphate. So we have those two different backbones, but we're still using our complementary base pair rules. And so this area where we're making the messenger RNA is called that DNA-RNA hybrid region. All right, so, and then as it makes it, we have that strand of nucleotides coming out from that. Now, we started at the promoter region, we latch on to the DNA, and then we don't just follow the DNA to the end of the strand. Eventually, we are going to reach a different sequence of nucleotides that is known as a termination signal. And that termination signal is going to tell the RNA polymerase to let go. So we initiate, we bind to the promoter region, we elongate using our complementary base pair rules, and then we terminate at the termination sequence. Notice, as we said, the DNA is completely unchanged from that process, but now we have a long strand. I want to be a little careful here. Immediately after this process, so let's write this here. I have a long strand of RNA nucleotides. Period. That is all we have. Once we have this long strand of RNA nucleotides, the strand is modified. Immediately, there are some changes that are made to it. Some sections will be cut out and put back together. It will be modified, and once it is modified, and again, in the nucleus, oops. Once it is modified in the nucleus, it then becomes our messenger. Oops, that's not how you spell messenger. RNA.
Can you scroll up a little bit because the words are cut at the bottom?
Here, I'll sneak it in there. You see it there?
Yep, thank you.
Okay, perfect. So again, at first, when you're done, you just have a strand of RNA nucleotides, but that strand is then modified. It is processed in the nucleus, and it becomes our messenger RNA. And once we have that messenger RNA, the messenger RNA, and again, once you've written out messenger ribonucleic acid once, we can then abbreviate it. mRNA is then able to leave the nucleus. And how would you leave the nucleus? How would something like this get out of the nucleus?
Through the pores, you mean?
Yeah, there you go, through the nuclear pores. So there you go. Once we make that messenger RNA, the messenger RNA is then able to leave the nucleus out through the nuclear pores onto the rough endoplasmic reticulum, which remember, we said is right outside the nucleus and sometimes actually attached to the nucleus. So as soon as it leaves the nuclear envelope, that messenger RNA goes straight to the ribosome, straight to the rough endoplasmic reticulum, so that we can start the process of translation. All right, so any questions on those 3 steps of transcription? All right, excellent. Now, we do have an issue, all right? Again, remember, messenger RNA is the language of nucleic acids. Right? Just like English is a language, and English as a language has 4 letters in its alphabet. How many letters are in the alphabet of the nucleic acids? Or RNA, more specifically? Come on, I know you know. 4, there you go, excellent. All right, there's only 4 letters. And they're A, C, G, and U. These need to code for our specific, so there are 4 letters, 4 nucleotides. They need to code for our amino acids. And how many amino acids did we say there were again?
4. Yeah.
How many amino acids?
20.
There's the magic word, 20. There are 20 amino acids, right? So what that means is A, does A just stand for 5 different amino acids and we can just pick any of those 5 to put in the sequence when we're making a protein? No, as we talked about, right, if you just change 1 amino acid in a sequence of thousands, like in sickle cell anemia, you can have misfunction of those proteins. So obviously it can't be a 1-to-1 ratio. Now, luckily, we can do math. 4 squared, right? If we primed 4, where's my little up arrow? Oh, there it is. What is 4 squared equal?
16.
16. That would almost be enough, but not enough. We need 20. So instead, what we can do is we can do 4 to the 3rd power. 4 to the 3rd power is what?
$64.
There you go. It's equals 64. 64 is more than enough options. So what that means is that when we're coming up with the translation of nucleic acids to amino acids, we are going to use a sequence of 3 nucleotides. And that sequence of 3 nucleotides will equal 1 amino acid. So again, for instance, I could have 1 AAA, and that's going to equal a specific amino acid. Or GCG is going to equal a specific amino acid, and so forth and so on. And we have the special name for these sequences of 3 nucleotides, and the name for this special sequence of 3 nucleotides is a codon. Now, any textbook worth its salt, and yours is going to have a pretty picture like this, a table like this, that is going to help you to figure out what those are. Right? So, for instance, G, your first letter's here, U, your second letter's here, C, your third letter's here, G, U, C. This particular codon codes for the amino acid valine. All right? And so on and so forth. Right? GGU, on the other hand, codes for a glycine, and so on and so forth. Now, here we see there are 20 amino acids. Obviously, as you can see, with 64 potential codes at any 20 amino acids, there is some replication on some of these. But I also told you you don't need to memorize your 20 amino acids. So you don't need to have your 20 amino acids memorized, but you do need to be able to read a chart like this or something similar to it. However, if you do notice on this chart, there are 4 highlighted codons. And that isn't by accident. Right? One of them happens to be highlighted green. And what does green usually mean?
Go.
Green means go. I know most of you are stuck at home and haven't driven in a while, but the good news is green still means go. And in fact, the one green one is what is known as the start codon. The start codon, if you notice, is AUG. That is going to be our start codon where we're going to start the process of translation. But it also codes for a specific amino acid. And that specific amino acid it codes for is methionine. So when we start our process of translation, we are always going to start with AUG, our start codon, and that means the first amino acid that is always brought to the party is methionine. That you have to have memorized. Notice there are also 3 red codons that have been highlighted on this. And what does red usually mean?
Stop.
Stop. In fact, it shows it right there when you look on the chart. These are what we call our stop codons. And guess what you do at a stop codon?
Stop.
You stop. There you go. See, it's not all challenging. Exactly. You stop the process of translation. And notice that stop codons, and there are 3 of them, UAA, UAG, and UGA. Those 4 codons, AUG, UAA, UAG, UGA, those you must have memorized. You must know your start and stop codons, not just what they do, but what their letters are and what they do, of course. All right, so those you have to have memorized. Anything else you should you'll have a chart or something like that that you'll be able to read off of. All righty. Questions on that?
Could you give an example of how you would go about reading it?
Yep, I will do that. We will exactly do that. We will absolutely go through this process together.
Gotcha.
All right, and talk about and see how these things work. But what I want you to understand is, again, remember translation is about changing the language. And this chart here is how we change the language, how we go from a language that only has 4 letters to a language that has 20 letters. This is how we get from nucleotides to amino acids. Okay, and if we understand this part, then we can actually see the process of translation and actually describe the process of translation in detail. Okay? All right, before we can do that, there is one other piece of business we have to do, actually more specifically 2 other pieces of business that we need to do, and that is to talk more about our RNA. Right, as we mentioned, we've already talked about messenger RNA. Messenger RNA is the information, the temporary copy of that information that is being presented to the ribosome to make our sequence of amino acids, to make our protein. But it's not the other, pardon me, blah, blah, blah. It's not the only RNA that is involved in this process. There's other RNA involved in this process. In fact, 2 other pieces of RNA. One is transfer RNA, which once you write it out once, you then can abbreviate tRNA. And the other is ribosomal RNA, which once you've written out once, you can abbreviate rRNA. So before we can talk about translation, we have to talk about these 2 molecules as well. Let's start first with transfer RNA. Transfer RNA is by far the smallest of all the RNAs. Right? As we mentioned, a messenger RNA, if a protein can be made of, and let's use simple numbers, if a protein is made up of 1,000 amino acids, how many messenger RNA nucleotides do we need to make that 1,000 protein, 1,000 amino acid protein. It's okay if you have to take your socks off to do the math. 1,000 amino acids. So how many codons would you need?
Is it like 20,000 or no?
Well, you would need 1,000 codons, but how many nucleic acids are in a codon? You guys are overthinking this. How many nucleic acids do you need to code for one amino acid?
4?
3.
Oh,
3, right? These 3 code for this amino acid. So if I had 1,000 amino acids, how many nucleotides would I need to code for that?
3,000.
Yeah, 3 times 1,000. And what's 3 times 1,000?
3,000.
3,000, right?
Oh.
3,000. You would need 3,000. Although technically, you would also, again, one amino acid needs 3 nucleotides, right? So 1,000 amino acids would need 3,000 nucleotides, but you would also need a start and a stop. So technically, you would need 3,006 nucleotides to be able to do that. Does that make sense? Wasn't the point I was trying to make, but hopefully that makes some sense. The point I was trying to make is that a messenger RNA is going to be made up of thousands upon thousands of nucleotides. So transfer RNA only being made up of 80 is an incredibly small molecule compared to messenger RNA, right? So it's the smallest of all of the RNAs. And it has a very specific job. It basically has 2 functions. Its job is to bring the amino acids to the party. So I'm gonna draw this symbolically first. We will look at the secondary and tertiary structure of it, but usually this is drawn symbolically. And when we draw this symbolically, on one end, there is a binding site where it can bind to an amino acid. And at the other side, it is going to have a sequence of 3 nucleotides. And the sequence of 3 nucleotides, let's go ahead and change the color to make it brown, is what is referred to as the anticodon. And this anticodon is going to allow it to bind to, oops, too far, is going to allow it to bind to the messenger RNA, match up with the messenger RNA. So its job, and I have to move my picture, we'll look at the picture of it in just a second, is to use that anticodon to match up with the messenger RNA. Here's the pretty picture that shows what it looks like. Here we actually see, notice here's our 2-dimensional structure, right? The same way we talked about the structures of proteins, we can talk about the structures of nucleic acids in a similar way. Here we see the single sequence of nucleic acids, but here we see the hydrogen bonds that form kind of this clover leaf shape. So this 3 clover leaf shape is that. But when it folds up into its 3-dimensional shape here, that we see here, and then like I said, symbolically, it's typically shown as a cylinder or a bent cylinder with a binding site for an amino acid at one point, which is what is up here. And down here is the anticodon. Yep, the anticodon is what is going to, the transfer RNA binds to the messenger RNA, matches up with the messenger RNA, absolutely. And like I said, I promise you we will go through this whole sequence step by step, and in fact, we'll do it twice. I'll do it once with my amazing, beautiful drawings, and then we will do it with all the pretty pictures from the textbook as well. All right. The third RNA molecule is ribosomal RNA. Now, you don't actually find ribosomal RNA by itself. It's never, let's say it that way, by itself. Instead, it binds with proteins to form our ribosomes. So basically, a ribosome is made up of proteins and RNA and ribosomal RNA. So, oops, hold on. I want to move that over. Ribosomal RNA and proteins come together to form a ribosome. A ribosome actually has two parts. These two parts are a large subunit and a small subunit. I'm going to cheat and draw this here. This we will call our small subunit. Our small subunit has one specialization. And that one specialization, and we'll draw it as a red circle, is it has a spot where our small subunit can bind to messenger RNA. So our small subunit is able to bind to our messenger RNA. It has a binding site where it holds the messenger, holds on to the messenger RNA. Our large subunit, oh, I don't know what this is going to be. Red. Let's go back to brown. Our large subunit has, let's call it 2.5. I'll put 2.5 in parentheses. 3 specializations. And it had, or 3 binding sites. There's 1. There's 2. And there's 3. And these 3 binding sites are what are known as the E site, the P site, and the A site. So why are they called the E site, the P site, and the A site? Well, the A site is where we bring in our new amino acids. So that is why it's called the A site, A for amino acid. Our P site is where we are going to hold the growing polypeptide. So that is why it is called the P site. And the E site is the site where our transfer RNA is going to exit. And since this is where it exits, it is the E site for exits. And again, that's kind of also why I said 2.5 specializations. Nothing stays in the exit site. The exit site is just a door you exit out of. So it's not really a binding site so much as a site that once the transfer RNA reaches this point, it is released from this point. So that's why I kind of hedge my bet with 2.5, but really 3 binding sites, the A site, the P site, and the E site. And again, I've done an amazing job of drawing this, but let's look at the pretty picture from your textbook. The job of the ribosome is to bring the transfer RNA and the messenger RNA together using their codons and anticodons. And like I said, it has those 2 binding sites, the A site and the P site, but it also has that exit site as well. And again, if you don't like my art, here is the pretty picture from the textbook. Here, again, we have our small subunit with its special binding site to hold on to the messenger RNA. Here is our large subunit, and this large subunit is where the transfer RNAs are going to be located. The transfer RNA that brings in the new amino acid is located here. The transfer RNA that is holding on to the polypeptide sequence is located here, and this is where our transfer RNA is going to exit. All right. Questions on that? All right, we have all the pretty pictures then. We have all the parts. So what we need to do now is put these pieces together and see how this process works. All right, to be able to do that, like I mentioned, I'm going to go through it twice. The first time, and again, you got to give me a break. This is the first time I've ever done it this way, because remember, we were in the classroom last semester when I did that. I will do my best to draw this and describe this on the whiteboard. We'll go through the process together. Hopefully it'll make some semblance of sense. And then if that works, then after we do that, then we will go through it again with all the pretty pictures and all the pretty words from the outline and from your textbook. So as I said before, when we go through it the first time, for this first time, I really want you to put your pens and pencils down, just lean back in your chair, just watch and listen, enjoy me struggling through this process of using this medium to be able to do this. And we'll go through it that way, and then we'll go through it with all the pretty pictures and words, and then you can write till your heart's content of that. I'll remind you also that you have some great resources online with the interactive physiology and other things that do a good job of describing this process as well. So if this doesn't make a lot of sense to you, then I will say that your resources online can really be helpful for this as well. All right, so before we dive into that, this is a perfect place to take our first break. So let's go ahead and take our first break. That'll give me some time to get set up as well. And what time is it? 9:06. So let's come back at 9:16, and at 9:16 we will restart. All right. So any questions before we take our break? All right. I will meet you back here in 10 minutes. So let's go ahead and get started. Like I said, this is going to be new for me, so we'll play with this and see how easy or hard this is and try to make some sense of this. But the important thing, no matter how sloppy this is going to be going through it, it's more important to listen to the words that I'm saying and not worrying too much about me fumbling with the artistry of this to make sure that we understand what's going on. So we're going to go through this process first together. So sit with me as we struggle through this, and then we'll see if we can make some sense of all of this. And then we'll go through the process again with all the pretty pictures and all the pretty words to make sure that you guys master it and understand it. What we have here are the plain pieces that are going to be involved in the process of translation. In translation, basically there are 4 things that need to come together. The first is our messenger RNA, and that's what we have here, a nice big strand of messenger RNA that has come from the nucleus. Our ribosome, which has 2 parts, a small subunit, which remember has a binding spot to bind to the messenger RNA, and our large subunit, which has 3 binding sites for our transfer RNA. And that's what we have over here. And remember, our transfer RNA is going to have 2 components to it. It is going to have an amino acid that is going to be bound to one side, and then it's going to have an anti-codon that is going to allow it to match up with our specific codon on the messenger RNA. All right? So the first thing that has to happen is these things all need to come together, and they all need to come together on, let's not so theoretically refer to as maybe a beginning point or maybe a starting point. What might a good starting point be? If only our messenger RNA had codons that coded, and one of them might have something to do with beginning. There you go, the start codon. And what was the start codon again?
AUG.
AUG. So notice what's going to happen here is our small subunit of the ribosome is going to start looking down the messenger RNA until, lo and behold, it finds, let's go with brown, the start codon. Notice here is the start codon. So what's going to happen is that our small subunit is going to come and bind to the messenger RNA at that start codon. when that occurs, our large subunit, oops, no, don't want to grab that part. I want to grab this part. Our large subunit is going to come and bind to it as well. And when it comes and binds, remember it has its 3 binding sites, the E site, the P site, and the A site. And so the 4th thing that is going to come in is our transfer RNA. Now, I'm going to leave this transfer RNA over here right now and make a newer version of it right here. So we have our transfer RNA that is going to come and bind in this site. But what you have to remember is not just any old transfer RNA is going to be able to come in here and bind. Only the transfer RNA that has the correct anti-codon. Our start codon is AUG. So what would the complementary RNA nucleotide be for A? What's complementary to A?
Would be
U, excellent. What is complementary to U?
A.
And what is complementary to G?
C.
C. So notice in this case, whatever that codon, that anticodon is, and in this case it is UAC, that is the anticodon that allows this transfer RNA and only this transfer RNA to come into this binding site and bind with the start codon. Now, we know that this, like all transfer RNAs, our transfer RNA is going to bind to an amino acid. It's going to have an amino acid bound to it. However, what's special about the start codon is, do we actually know which specific amino acid is going to be here? What does the start codon AUG? AUG is the start codon, but remember we also said it coded for an amino acid. Methionine, excellent. And I don't have the space to write methionine in here, so I'm just going to abbreviate it MET. So these 4 things come together. The small subunit of the ribosome, the large subunit of the ribosome, the messenger RNA, and our first transfer RNA. Those 4 things come together. And they all come together on one spot. And that one spot they come together is the start codon. All right, so far so good? Excellent, super exciting, but notice our A site is open. And A, remember, stands for amino acid. So what that means is a new transfer RNA can come into this A site. Now, can it be any transfer RNA? Can any transfer RNA come into this site?
No.
No, it has to be specific. Again, it has to be the specific, it has to have the specific anticodon to the codon sitting in front of it. And the codon sitting in front of it is CGC. So if the codon in front of it is CGC, what would the correct anticodon be?
G.
G.
GCG, exactly, perfect. Excellent. It would be GCG, perfect. So GCG, the transfer RNA with GCG would come in, and when it came in, it would bring a very specific amino acid with it. Now, off the top of our heads, memorized, do we happen to know what amino acid it is? Anyone have it memorized? I don't have it memorized, so don't feel bad if you don't either. Hold on, no, that's not what I'm trying to do. I'm trying to do that. I want to squish that a little bit. Excellent. No, but if we cheated and went back to the previous page, would we be able to figure out what it is? Yeah, we could figure out what it is. But I don't care. We don't care right now. We could look it up if we wanted to. We don't care. We just know it's the second amino acid. right? Amino acid, there's the second amino acid in this spot that is going to come in. We don't care what it is off the top of our heads. We just know it's going to be an amino acid. And in fact, I'm going to undo that now. Let's actually make it, I'm going to just go AA2. I don't think that's going to fit in the space, but so we'll make it smaller.
You can decrease the size.
Yep, that's exactly what I was going to do. But now I'm like using 3 different sizes. But hey, like I said, this is the challenge of it. All right. So far, so good. We have 2 transfer RNAs with 2 amino acids in our 2 binding sites. There's no room for any more amino acids to come in, no room for any more transfer RNAs, but now the work can be done. And let's start with the simple rule first. Once we have our 2 transfer RNAs in here, what we are going to do is 2 things. We're going to make a bond, and then we are going to break a bond. All right, that's how I remember. We make a bond, and then we break a bond. Now, let's think about this. What is the process, again, by which we make a bond? I'm going to use purple, and I'm going to make it smaller. Perfect. What was the process we used? There we go. Excellent. We make a bond using a process we call dehydration synthesis. Remember, with dehydration synthesis, we are drawing water out. And so specifically, where we make our bond, we make a bond between the 2 amino acids. Of course, remember when we form that covalent bond, remember we called that a peptide bond. So we're going to form a peptide bond between those. And because we're using dehydration synthesis, in that process, we draw out a water. So what ends up happening, and let's draw it here, and I'm going to use the big line and use black to form that peptide bond between the 2 amino acids. And when that occurs, remember as a result of that, and I'm going to have to do this teeny now. So there and here. As a result of that, we get a water. So blue, tiny. When we form that bond, we get a water as a result of this, right? Because we're using dehydration synthesis to form that peptide bond, and that produces a water as a result of that. All right, you guys with me so far?
Golden.
Excellent. So again, we have now made a bond. Check. We have done that. Now our job is to break a bond. And if we are going to break a bond, what is the process that we use for that again? Hydrolysis. Excellent. To break a bond, we use a process we called hydrolysis. See, I don't just pick the things that we talk about just randomly out of a hat. We do this for a reason, absolutely. We are going to use the process of hydrolysis. And remember, hydrolysis is where you use water to split, to separate or break a bond. Oops. Say it this way. And more specifically, let's be specific, to break the bond. And what we are going to do is we are going to break the bond between the first amino acid, well, let's say it this way so it's more specific. To break the bond between the amino acid and the transfer RNA in the P site. So what we're going to do now is we are going to use that water to break this bond. that didn't work. Let's make it a little bigger. 24. X. I like that. That one should work. And we're breaking this bond right here between the transfer RNA and the amino acid in the P site. So notice step 1 was to make a bond. We've done that. Step 2 was to break a bond. We have done that. We've now done all the work we can do at this location. All right? With me so far? All right. Complete, utter silence tells me, yes, I completely understand this process. Please continue. Perfect. Excellent. So now that we've done all the possible work that we could possibly do, then what we are going to do is do what you should be doing, well, or not should be, because you shouldn't be doing it right now, but back in ancient times, what you used to do on a typical Friday night. And on a typical Friday night, you were sitting there looking at your phone, and when you saw something you liked, you swiped right. And that is exactly what we're going to do here. Now we are going to swipe right. The entire ribosome is going to swipe right. It is going to move to the right. So, and again, this is where this is going to get a little tricky. So let me grab all of this stuff and sneak it over here and sneak this over here. And I think that's enough out of the way. So what's going to happen is, and again, this all happens at once, my small subunit shifts down one spot. My large subunit, no, not you. My large subunit shifts down one spot. And notice as it shifts down one spot to there, I shifted too far. Perfect. What has happened is my transfer RNAs have moved. The first transfer RNA is now in the E site. And what does E stand for again? What does E stand for? Exit. Exit. And so guess what the transfer RNA, especially because it's no longer being held to the amino acid, guess what the transfer RNA does?
Leaves.
It exits. There you go. It sallies forth. It exits. It can scamper off, where in fact it could go back to an enzyme and actually get a new amino acid put on, and it could be used again in that process. Notice the second one is now in the P site, holding our growing polypeptide. And now we have room for a new transfer RNA to come in. And that's what's going to happen. A new transfer RNA is going to come in. And not just any transfer RNA, but the one specific. This is a little too far in my way. Move this back a little this way. Cheat and move that way. Make this a little bit bigger. Excellent. And based on our complementary base pair rules, what is the one and only transfer RNA that can come in here? What anticodon does it have to have? Well, the codon is CAA, so what is my anticodon? GUU. Perfect. Excellent, 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 excellent. Oops. GUU. That transfer RNA comes in, and it is going to be bound to an amino acid. Do we have to have that amino acid memorized off the top of our head? Nope. But could we look it up if we wanted to? Absolutely. So there's my amino acid, and everything's getting a little squished in here, but we get the idea. And as we talked about, this one is going to be amino acid 3. Perfect. All right. Excellent. So my ribosome shifted right. Space opened up for a new transfer RNA. A new transfer RNA came in. I have a transfer RNA in the A site, a transfer RNA in the P site. What's going to happen now?
Is everything gonna shift towards the left again?
Well, first of all, it shifted right, not left. But
I'm sorry. Yeah.
before we can shift right, we have to do some work. And what is not the one, but the two pieces of work we have to do?
Make a bond, break a bond.
Mm-hmm. Absolutely. We need to make a bond and break a bond. Excellent. Which one do we do first?
Make a bond.
Make a bond. Where do we make that bond?
Wherever it fits, wherever it coincides.
True. And where is that? Again, that's inaccurate, but again, we want, especially on the exam, we want to make sure we're precise in these definitions. Excellent. Between the amino acids, between the second and the third amino acid, right? That is where we're going to make a bond. And of course, what process are we going to use to make that bond?
Dehydration synthesis.
Dehydration synthesis. And what do we get as a result of dehydration synthesis?
Water. Bond.
We get a water. Excellent. And when we get that water, we can then use that water for our second process. And what's the second process?
Hydrolysis.
Hydrolysis. And where are we going to break the bond?
Or the second and the first.
Sort of in the second, but right. So again, that's why I changed my definition over here. Where we break the bond is between the amino acid and the transfer RNA that is in the P site. That way we don't have to worry about if it's the second or the third or the 850th. We just know that here in the P site, we use that water to break that bond between the transfer RNA and the amino acid in the P site.
Now, is it always gonna break in the P side?
Yes, it is always going to break in the P site. It always breaks in the P site. So there you go. We shifted, we brought in a new transfer RNA, and we did our two pieces of work. We made a bond and we broke a bond. So what's going to happen now?
Exit?
Exit.
True, but to get it to exit, what does the ribosome have to do?
Right.
Shift to the right. Excellent. So once again, the whole thing shifts to the right, right? Swipe right. Don't pretend you don't know what I mean when I keep saying that, right? So this thing is going to swipe to the right, shift to the right. Our second transfer RNA is now in the exit site. So, whoops, wrong thing. So again, it leaves. Whoa, what happened there? There we go. It leaves, right? And takes its anticodon with it. Our second one is now in the P site. And we now have a new open A site. So this process continues again. New transfer RNA. Oh, don't want it to be blue. New transfer RNA, and again, can it be any old transfer RNA? These questions should be getting easier because it's the same process over and over again.
Exactly, that specific anticodon would go.
And what would the anticodon be in this case?
AGG.
AGG, perfect, excellent. And it is going to bring a new amino acid with it. As it brings that new amino acid with it, we are then going to do some work. What's the work we're going to do? Again, the question should be getting easier. What's the work we're going to do?
We're gonna form a bond, or?
Yeah, and where do we form a bond?
Between the P and the A?
Between the amino acids in A and P. Excellent. Again, be specific in your wording of these things. What process do we use for that? Again, these are all the same processes again and again. These questions should be getting easier. What process do we use to make the bond?
Dehydration?
Dehydration synthesis, which gives us what as a result? What do we get as a result of dehydration synthesis? A water. Excellent. Once that water is formed, what do we use the water to do? Oh, don't want that. We use it to break the bond. Excellent, right? Again, this process continues again and again. I keep saying it again and again because I want to make sure we understand it again and again. We're going to keep going again and again and again and again and again, right? And this process continues step by step by step by step. Notice it's going to shift again, and it's going to shift again, and it's going to shift again, and it's going to shift again. It's going to keep shifting, and this process is going to continue thousands of times until something special happens. Let's cheat and fast forward a little bit in the process. This process continues again. No. And I don't think I have the right number of amino acids here, but you guys get the idea. So I'm cheating a little bit and we're fast forwarding a little bit in the process. But hopefully this is getting easier as we talk about it again and again and again. There, so bond, bond. There. So again, what has been happening is step by step by step, we shift to the right. A new transfer RNA comes with the specific anti-codon that complements and matches the codon. It brings a specific amino acid. When it brings that specific amino acid, we then make a bond between the amino acid that is in the A site and the P site using dehydration synthesis. That gives us a water, and we use that water to break the bond between the transfer RNA and the amino acid in the P site. That then allows the ribosome to shift to the right again, and the process continues. Whatever was in the P site now is in the E site and can exit. Whatever was in the A site is now in the P site with the growing polypeptide, and now we have room for a new transfer RNA to come in. And again, that is going to be what happens. A new transfer RNA is going to come in using the same complementary base pair rules that we've been dealing with before. So let's down a teeny bit. However, look at the codon we are at now. What do you notice about this particular codon?
Stop one.
It's one of our stop codons, exactly. UGA is one of our stop codons. What were the other two stop codons?
UAG.
UAG and?
UAA.
UAA, excellent, perfect. So we're going to bring in a transfer RNA. This transfer RNA is going to have the correct anticodon. It's the only way that it can come in. So what would the correct anticodon be for UGA?
CU.
Now we don't use T, so it would be
ACU.
ACU.
ACU. Excellent. That is what is going to come in this particular transfer RNA that matches this codon. But here's where things are going to be a little different. Whereas every other transfer RNA that has come in has brought an amino acid, if you remember correctly, when we looked at that chart, did the stop codons have amino acids associated with them?
No.
No, they didn't. So what's special about these transfer RNAs, what actually causes this process to stop, is that these transfer RNAs don't bring an amino acid. What they actually bring is a water. So instead of bringing an amino acid, what these transfer RNAs associated with the stop codons do is they bring a water with them. And guess what that water is used to do? What have we been using water to do up to this point in time?
Breaking bonds.
breaking bonds, and that is exactly what happens. This water is used to break the bond between the transfer RNA in the P site and the amino acid in the P site. And when we break this bond, notice our string of amino acids is no longer being held in place. It is now completely free and released, and it can float off, and now is going to be shipped to the Golgi apparatus. There's
Question.
nothing holding it in place anymore. Yes.
So all the other ones before this, they're all still connected, even though they exit, correct? They're still
Remember,
connected to each other?
what exits is the transfer RNAs. All the amino acids have peptide bonds that are holding them together. So this strand forms and stays attached. And it's always been anchored to a transfer RNA that is keeping it inside of the ribosome. But when this one brings a water, it breaks that bond and it's no longer connected. And so now this whole strand of amino acids can float off and be moved to the Golgi apparatus and become a fully functional protein. In fact, at that point, there's nothing holding anything together. And at this point, the entire ribosome, the entire structure comes apart. So the large subunit lets go, the small subunit lets go, everything lets go, and the process ends.
Quick question. Before that, every time the transfer RNA has exited, it's no longer the what is it called? You said it's not going anywhere, and it's going to form a protein once the bond is broken, correct? But before that, it's still intact, even though it's not anchored to anything.
Okay, I see what you're saying. Yes. So yes, so remember, our transfer RNA is bound to an amino acid.
Yeah.
Remember, when it reaches the P site, we use water to break that bond.
Mm-hmm.
Now, our amino acid is still attached to other amino acids by peptide bonds, but the transfer RNA, like this one over here, is now completely free. So it's no longer attached to the amino acid, it's no longer held to the chain, so it can freely float off and do its own thing. And in fact, what it's going to do is go find a special enzyme, and that special enzyme is going to put a new amino acid attached to it. So it can get a new amino acid attached to it and could actually be used again in this one or some other protein synthesis process. So our transfer RNAs can be reused, but they need to go get a new amino acid first. But they're no longer attached to this strand that we're forming. Did that answer your question?
Yep.
Okay, perfect.
Thank you.
Now, so any other questions on this process? All right, so at this point, remember this ribosome breaks off. This process stops. Our sequence of amino acids now gets shipped to the Golgi apparatus to get converted into a fully functional protein. But notice one last thing. Did we change the messenger RNA in this process? No, the messenger RNA wasn't changed at all in this process, which means it can actually be used again. Which, A, if you think about it, makes sense, because if a cell needs a protein, is it likely to just need one copy of that protein? No. It can be read, a single strand of messenger RNA can actually be read thousands of times to be used to make thousands of copies of that protein. And what's really cool about this process is if you think about it, because the ribosome moves down the messenger RNA, as it shifts and moves down the messenger RNA, a new ribosome can actually come and bind to the beginning and start as well. So it's actually possible for a single strand of messenger RNA to be being read by dozens of ribosomes at the same time. Your book actually has a beautiful picture of this. I'll show you a little bit later, where it actually, you can see that. Using an electron microscope, we see a single strand of messenger RNA that has a dozen ribosomes all moving down them together, all making copies of the DNA at essentially the same time. All right. It wasn't quite as horrible as I feared. It wasn't great, but it's easier to do it when I can erase and move things on the board, but that wasn't horrible. All right, let's look at the pretty pictures of the textbook and do the entire thing again.
Yay.
I know, exciting stuff. But hey, look, think about it. This is the entire subject of what we're talking about today in class. Does that make it a pretty important process?
Yeah.
Are you almost guaranteed to have at least one of those 3 essay questions and possibly more that could come up associated with this? Yeah,
Yeah.
again, I don't think I'm giving away any trade secrets to let you know that there is going to be a protein synthesis essay on the exam, right? I mean, anything we spend this much time in is something that is definitely going to be on the exam. And again, that's what I love about this. I can say that because I know you still have to learn this process. So that's the easy part about this for me. That's what I love about this class. This class is hard, so I can tell you, hey, there's going to be a protein synthesis question on the exam. Do I have any idea what question you're going to get randomly assigned? No, no idea at all. But are there a bunch of questions in there for you on this stuff? Absolutely. All right. So let's make sure we understand that process so we can be correct and accurate in describing this on the exam. Now, we are going to use this really simple, pretty picture because it's very simple and basic and walks us through it. But what I will tell you is your textbook actually has a really beautiful illustration that I'll show you at the end of this that does a really great job. So the other thing that I will tell you is aside from the other resources that you have, your PhysioX, the interactive physiology, all of those things that are in there, your textbook actually does a really, really good job of describing this process. So if, again, the way I'm describing it doesn't make sense, sometimes reading it in a different voice, having someone else use their language to explain it, can sometimes help this information to click. So this is one of the areas where your textbook is really excellent. And it has a great illustration that shows it as well, which I'll give it to you and show you at the end of this. But here's a simple version, but it still goes through all of the important parts. Notice, as we talked about, there are 1, 2, 3, 4 things that need to come together. And here, let me bring up my annotation so I can get my highlighter. The messenger RNA is thing 1. The small subunit is thing 2. The large subunit is thing 3. And the transfer RNA is thing 4. Now notice they didn't bother drawing us an exit site on this simple version. We just have the P site and the A site because those are the binding sites. Those are the important things where they stay. But it still has it in there. And notice the other key is these 4 things that come together, all come together on that start codon. So here we have that start codon, AUG, our transfer RNA with the correct anticodon. And if AUG is the start codon, then UAC is the anticodon. The large subunit and the small subunit come together. And our transfer RNA brings our first amino acid. And what do we know that first amino acid is always going to absolutely positively always be?
Start code.
Methionine, absolutely, right? So methionine. So again, it always brings methionine. Now, does that mean that every single protein in your body, the very first amino acid on every single protein in your body is methionine? Well, it turns out no. It is when we make it. But remember, when we make these, we just get a string of amino acids. And this string of amino acids goes to the Golgi apparatus. And remember, we talked about the Golgi apparatus cuts it, folds it, and puts the pieces together to make a protein. So typically, one of the first things the Golgi apparatus does to get it to fold up properly is cut off the beginning part of the sequence. And once it cuts off the beginning part of the sequence, that typically allows it to fold up into the shape that we want. So when we make the sequence of amino acids, methionine is always the first amino acid. But by the time it becomes a functional protein, it is usually cut off. So not every protein in your body actually has methionine as its first, but it was the first when we made it. All right? So these 4 things come together. Then a second transfer RNA comes in, again, using the specific anticodon to the specific codon on the messenger RNA, bringing a specific amino acid, which, again, we don't have memorized what CCG codes for, but we could definitely look it up if we needed to. And it is going to come in, bringing the next amino acid in that process. At that point, all of our transfer RNAs are in place. And at this point, all right, we do our work. And doing our work is 2 things. We make a bond, and then we break a bond. The first thing we do is we make that bond, and we form that bond, a peptide bond, between the two amino acids. We use dehydration synthesis for that. I don't know why I'm having a problem saying synthesis. Dehydration synthesis, I haven't had enough caffeine. Dehydration synthesis is what is used to make that bond. And from that, we get a water. And then that water is used to break the bond between our transfer RNA and the amino acid in the P site. So we form that peptide strand. We break the bond with the original transfer RNA. And then everybody swipes right. We shift to the right. And when we shift to the right, the first transfer RNA exits the exit site. Our second one is shifted into the P site, where it's holding the polypeptide strand. And now our A site is open for a new transfer RNA to come in with its specific anti-codon to match the codon. Once there, make a bond, break a bond, swipe right. Bring in a new transfer RNA, make a bond, break a bond, swipe right. And again and again and again, hundreds, if not thousands of times. This continues until we reach the stop codon. And what are the three stop codons again?
UGA,
UGA,
and UAA.
Is it UAA or UUA?
UA.
Okay, UAA and UAG. Those them? Yep. Is that right? Are those right?
UGA.
What? UGA, UAG, UAA? Are those our three? How many times do I have to ask? Come on. Are those our?
Yeah.
Okay, excellent. Perfect. Excellent. Now, what's special about the transfer RNA that comes in with the anticodon for this? What is special about this one that stops the process? Right? It does not bring all right. What's special about this? Exactly. It does not bring an amino acid. Instead, it brings a water. And so that water is able to be used to break the bond. And once it breaks the bond, then as we talked about, the whole thing disassembles. Our polypeptide is complete and released. And once released, again, it is going to travel to the Golgi apparatus or the Golgi complex. Remember, those two terms are interchangeable. And remember, the other thing about this too is that our messenger RNA is not changed by this process. So it can be read, in some cases, hundreds, if not thousands of times. All right. Your book's got a couple pretty pictures. Here, as I mentioned, is that great figure from your textbook, where it really does an excellent job of talking about this process. Notice here we see the three sites. As I mentioned, it actually shows the enzyme, and they talk about how you bind the amino acid to the transfer RNA. You don't have to know that process, but just know that we have to put the right one on here. And it comes in step by step, right? New amino acid comes in, form a bond, break a bond, swipe right, and that process continues thousands of times until you get to that stop codon. And again, it brings a water to the party to break that bond and allow the process to end. And as I mentioned, here is the other pretty picture from your textbook where, again, because the mRNA is not changed and because the ribosome slides to the right along it, multiple ribosomes can bind to it and move their way down and let go, and it can be read thousands of times. And I know it's easy to look at an illustration and see how this might work, But as I mentioned, your book actually has this beautiful electron microscopy view. Using an electron microscope, they have found a single strand of messenger RNA, where as you can see, 2, 4, 6, 8, 10, like a dozen more and slightly more ribosomes are all moving down that single strand of messenger RNA to be able to make those many sequences of amino acids that are going to become the many proteins that the cell needs to function. All right. Questions on that? All right, excellent. Couple more things then. Here is a nice illustration that also describes this process. Notice here, instead of actually describing the process, we're actually doing the work. Here we have the template strand of DNA. Here is the messenger RNA that it would code for, and the sequence is the codons. These then code to our anti-codons of a transfer RNA, and if we had a table, we could actually then come up with the sequence of amino acids. So not only do you need to be able to describe this process, but you need to actually be able to do this process. If I gave you a strand of DNA, you should be able to tell me the sequence of amino acids. If I gave you a sequence of amino acids, you should be able to work backwards to give me the strand of DNA. That makes it. All right. We have one more issue that we need to talk about, because as we talked about, in your body, you have over 200 different types of cells. And those 200 different types of cells, remind me again, do they have the same DNA in them, or do they have different DNA in them? All the 200 different types of cells in your body, do they have the same DNA or different DNA?
It would have different DNA because they're producing different proteins, right?
That would seem to make sense, but then again, how then can those CSI people be able to identify you based on your DNA if every single cell in your body had different DNA?
So they have the same DNA.
They have the same DNA. Every single cell in your body, with the exception of the sex cells. Remember, sex cells are being used to make other people. Those don't count. But every single somatic body in your cell has the exact same DNA. But you hit on the big key problem with that. Every single cell in my body has the same DNA, but every single cell in my body does not make the same proteins. Right? My toenail cells are making toenail proteins. My liver cells are making liver proteins. My eyeball cells are making eyeball proteins. If every single cell has the exact same DNA, then how is it that I tell eyeball cells to make eyeball proteins and liver cells to make liver proteins and toenail cells to make toenail proteins? And the other issue is, maybe I don't want my toenail cells making toenail proteins all the time. Or maybe my eyeball cells need to make different proteins now and different proteins an hour from now. If every single cell has the same DNA, how in the world do we control not just what proteins the cells make, but when they make it? How is this whole process regulated?
By chemical signals and hormonal signals?
Absolutely 100% correct. The way this process is regulated is by chemical signals. And what's the primary chemical signals we are going to use to tell a cell which proteins to make?
Ooh.
Come on, you know. What system is specialized for communicating with chemicals? What organ system is specialized for communicating with chemical signals?
Is it because of neurons?
Endocrine system, the endocrine system. And what do we call the chemicals made by the endocrine system?
Hormones?
Hormones, exactly.
I thought you were going somewhere different with that.
Too easy? Hormones are too easy? Yeah, hormones. Absolutely. Chemical signals, primarily hormones, is how we tell cells which proteins to make and when to make them. So this whole process of protein synthesis is controlled by hormones. And in fact, when we get to the endocrine system in 431, we will actually see the exact mechanism by which a hormone tells a cell which protein to make and when and why. All right? Questions on that? All right, excellent. So like I said, the only part of this, like I mentioned, that is going to be tricky for you, if at all, would be to, oops, no, that's what I want. Well, actually, let's cheat and go back to this. This is what I want. The only part of this that could be challenging for you as a student on the exam is this part down here. As I mentioned, with the exception of methionine, which you know is associated with the codon AUG, you do not have to memorize that codon chart, but you do have to be able to read one. So if I gave you a codon chart or a transfer RNA anticodon chart, you should be able to do this process of going from DNA to amino acid or going from amino acid to DNA. So that'd be the only part that you would need help with, would be some type of table. So you need to be able to read it. But like I said, you don't have to memorize all the codons. You just have to memorize those 4. You need to know what the start codon is and what it codes for. You need to know the 3 stop codons and what they code for. All right?
Can you give us an example of like a question that you would use?
Sure, like I said, so like this would be, right here, would be a perfect example of a test question. So question 1 on the exam could be, I could give you this strand of DNA, and if I gave you a chart for, I could ask you, all right, with this strand of DNA, give me the sequence of amino acids this would code for. So would you then have to figure out, all right, if this is my template strand of DNA, what is the messenger RNA that would be produced from it? Then what are the codons that would then match with the anticodons, and then which amino acid it would be able to form? Or I could ask it the other way as well. I could give you a sequence of amino acids, and I could ask you to give me the DNA, the strand of DNA, the template strand of DNA that would code for this. And should you be able to go from amino acid to transfer RNA or messenger RNA to DNA, and should you be able to code your way back from that? Yes, so you should be able to answer that question in either of those directions. If I gave you a chart, you should be able to go from amino acid to DNA or from DNA to amino acid. Okay? Yeah, the only thing I'd have to do is I'd have to give you a codon chart, because you don't have to memorize, you have to memorize the codon. I mean, you don't have to memorize the codon chart. So the only thing I would have to do is give you some type of chart. And that could be like this, right? If I gave you, oops, not, if I gave you that part of it, stop it. If I, hold on, let me clear that. If I gave you this, where these anticodons coded for this amino acids, and I gave you a series of these, then should you be able to give me what the sequence would be that would be from that? Absolutely. Okay?
Right. So we need to memorize the amino acids and how they translate to the anticodons so that we can basically tell everything else and vice versa.
You just need to know how to go from transfer RNA to messenger RNA for DNA. You don't have to know that AUU transfer RNA codes specifically for leucine, right? That part you don't have to know. I would give you that part of that. So I would give you that part of it, but you should be able to, using complementary base pairs, go from transfer RNA to messenger RNA to DNA.
If we're going backward from amino acid, we are going amino acid, then the anticodon, the messenger, and then the
template strand.
template.
And as we just pointed out here, if you give me the template strand of DNA, can you also give me the coding strand of DNA that goes with it? Yeah, absolutely. All right. Now, if we're going from DNA, oh, I like that question too. Let's go back. So this is the template strand right here, right? This is our template. Could I theoretically give you the coding strand? If I gave you the coding strand of DNA, then you would have to, from there, figure out what the template strand is. Then from the template strand, you would have to figure out what the messenger RNA is. From that, you would have to figure out what the transfer RNA is. And then with the chart, you would be able to figure out what the amino acid is. And you should be able to go this backwards as well. You should be able to go from amino acid to transfer RNA to messenger RNA to template strand, and if need be, you should be able to go to the coding strand as well. So absolutely, you definitely need to distinguish between template and coding. But if you know one, then because of complementary base pair rules, you should know the other. So absolutely, you should be able to go all of those steps if necessary.
Aren't we using the mRNA strand to read the chart?
There are charts that work that way, but I could also give you a chart of the transfer RNA. So that is, again, one of those places where you have to read the question carefully. If on the exam I gave you this chart, notice this chart is for transfer RNA anticodons. Whereas if we go all the way back here, no, too far, not enough, far enough. This is for codons. So yeah, you have to read the chart carefully because I could give it to you both ways. I could give you a codon chart like this, or I could give you an anticodon chart. So make sure you pay attention because they are going to be different.
Okay.
So read the question carefully and you should be able to answer it correctly. All right, great questions. Any others? All right, so then where are we? Oh, we're perfect on time. Excellent. So here is the game plan now. Let's go ahead and stop sharing for a moment. Hold on. Stop sharing. Come back to this screen. All right. So here is what is going to happen now. It is, actually, let's slide. Let's do this. Come back to the whiteboard. Even though this whiteboard is a bit of a mess, I'm going to take a picture of it just to save it and put it onto the, our, I don't know if it'll make any sense. I don't know if I'll help, but I'll post it onto our module. So that if this helps at all, so something you can recall to, I'll have that picture. But so I've saved a picture of it now. So now I'm going to go ahead and clear it. And let's talk now about your practice lab exam. All right. So it is 10:17 right now. You are going to have, it's 20 questions, and you are going to have 25 minutes to complete it. All right. Again, there's no video recording, no video proctoring. You don't have to show your ID. You don't have to do any of those things for this practice one. However, for the real one, you do. And again, that's the other thing that I meant to mention. I keep forgetting to mention. On the exam, for like I said, for the real, the two biggest errors that I saw when people were taking the exam is they did not scan their work area. So again, scan your entire work area and do it slowly. Right? Again, I'm the one watching these. I'm the one evaluating it. And you've got to appreciate that I've got to try to be consistent with everybody. So, but again, there's going to be some understanding. If you are constantly looking down to the right and you've never shown me what's down over there, then I don't know what you're doing. I appreciate that a lot of people look away when they're thinking about things. But if I don't know what's sitting next to you, then I don't know what that's going to be. If you look down there 4 times and you never showed me what's down there, then I may have to dock you points for that. If you're not scanning your area, remember you lose 10% right off the top. That means you have to get a perfect score to be able to get an A. Right, and that's very challenging. So make sure you show me your area. And if I know that area is clear, then I'm more likely to be able to be more lenient if you've looked down there 4 times. Right now, that doesn't mean you can bring something up once you've started. And again, I should point that out as well. For a longer exam, there will be random times during the exam where you are required to scan your area. You need to scan your work area slowly every time you're asked to scan. It's not just at the beginning and then you can ignore it the rest of the time. So make sure you scan your work area slowly every time, or you will lose 10%.
A quick question.
Yes.
When it's asking you to scan, like you said, multiple times, does the time stop while you're doing that, or it still continues?
It still continues, but that's why I've put extra time into the exam. The scan only takes a couple minutes. It doesn't even take a minute. It takes a couple seconds. It takes 15 seconds for you to scan your area. So again, at most you lose half a minute, but I've given you plenty of time. In the classroom, you typically get 1 minute per question for the lab exam. You're getting more than that for this. In the classroom, you typically get 2 hours for a lecture exam. You're getting 2.5. So you're getting more than enough time to make up for any of the technological issues that you have to deal with on this. The other issue, and again, most of you were pretty good at it, but there were a couple of you who did not do it, and it is important. You must scan your photo ID at the beginning. It doesn't have to be your driver's license, although if you want to use your driver's license, that's fine. It can be your student ID, but there's plenty of people where there was their head. That's not a photo ID. Again, I've never met any of you. I need to see a photo ID to make sure that the person taking this test is the person who's getting credit for this. So you have to show your photo ID. If you do not scan your photo ID, you will not get credit for the exam. If you don't show a photo ID, I don't know that it's you taking the test. And if I don't know it's you taking the test, then you can't get credit for the exam. So make sure you scan your photo ID at the beginning. It can be your student ID, that is perfectly acceptable. And if you don't want to show your driver's license, that's fine. But I have to see it has to be a picture ID that has your name on it, and hopefully it's in your Blockbuster card or something like that. I know none of you know what that is. I don't care. All right, so those are the important things for that on the real exam. You're not going to be doing the recording on this, but it will lock up your screen and do all of those other things that it will do for that as well. Remember, every single question has been a question on previous exam. Some of these questions are questions in your test bank, which means it is possible that you could see these again. So again, make sure the point of this is to give you an idea of what to expect from this. Now, what we are going to do is I'm going to make it available so that you can take it. You'll have the 25 minutes to take it, but then I also want to allow us to take our break as well. So it's 10:21 right now, 25 minutes for the quiz and then a little bit of a break. Let's meet back here at, let's see, 25 minutes. That's 45, 47. All right, we can meet at 11. So we will start class again at 11 a.m. At 11, so again, if you want to take your break first and then come take the practice quiz, you can do that. If you want to take your practice quiz first and then take your break, if you want to take a very small break, quiz, and then a small break, whatever, come back at 11. 11, we'll go through the quiz together quickly, make sure it makes sense, you understand what you did, you didn't do well, and then we will finish with whatever time that's left. We'll talk a little bit about tissues. All right,
Okay.
Questions on any of that? Yes.
Where is this quiz? I've been looking for it. I can't seem to find it on.
Because I haven't made it available yet because I've been talking about it. So let me do that right now. So let me stop sharing. No. All right, don't start it yet, but look now. Is the practice quiz there now?
Yes.
All right, then there you go. All right, perfect. So take your quiz now, take it at the end, whatever you want. But again, it's going to take about 20, 25 minutes. So take your quiz, take a break, and I will meet you back here at 11 o'clock. All right, any questions on that? All right,
Nope.
Good luck and have fun. The recording. Thank you. I didn't want to do that for the practice exam, but thank you. I did want to do that for this. All right, so excellent. Just to finish up, we have started tissues. We are identifying the 4 general types of tissues. We have identified them as being muscle, nervous, epithelial, and connective. Thank you for that reminder. I greatly appreciate it. All right. So muscle tissue can provide protection, right? But what else does it do?
Contract.
Support. Yeah, absolutely. It provides, it does protection, support, movement. It does all of those things by basically having a very special function in that it is able to contract. It is able to produce contractions. Excellent. It's very, very specialized tissue. And as we talked about earlier, it comes in 3 flavors. What are the 3 specific types of muscle tissue?
Cardiovascular.
Cardiac, excellent, cardiac. I see smooth. And what's the third? Skeletal, excellent. Excellent, so it comes in 3 flavors. All of them produce contractions and does things like support and movement and protection and all of that. Nervous tissue is another highly specialized type of tissue. And it's specialized for what? What did we say our nervous tissue is specialized for?
Internal communication.
Excellent. Specialized for communication. And how does it communicate again?
By sending signals, electronic.
Yeah, it produces an electrical signal. And remember, we called that electrical signal an action potential. Excellent. Those are 2 highly specialized types of tissues. So highly specialized that we need to know their tissue types. We need to know this basic information about them. But that's really all we need to know for this exam. Muscle tissue we'll talk about in much more detail when we get to the muscular system. Nervous tissue we'll talk about in much more detail when we get to the nervous system. But for this exam, you are just, other than, you need to know your foreign tissue types, you need to know general information about them, but you're not going to have to know any more detail about muscle and nervous tissue. This exam is going to be on epithelial tissues and connective tissues. So this exam is epithelial tissues and connective tissues only. So let's describe their functions. What is the function of an epithelial tissue? What do epithelial tissues do for us? Where do you find them?
Protected. Protect.
Excellent. One of the things they do is they protect us. Why? Where are they located?
Skin?
Well, the skin is one of the locations where we find it, but you're right on the idea. Epithelial tissues are superficial. They line all of the surfaces of the body, both inside and outside of the body. That allows them to provide protection, but it also does, like our skin is an important barrier, but it's not an impenetrable barrier. We want to be able to release material. We want to be able to absorb material. So it's also going to allow for absorption and secretion. And another important thing about epithelial tissues is epithelial tissues are also going to be used to form our glands. We're going to talk a lot about glands in this class, and epithelial tissues are what used to form our glands. Connective tissues, if epithelial tissues are superficial, our connective tissues are deep tissues. They're also the most diverse of all the tissues. And if they're most, well, I should say, let's do it this way. They're most diverse of all tissues in structure. And if they're the most diverse in structure, what else do you think that's going to make them?
Diversity function.
Yep. Of all the tissues in function as well. Excellent. They can also provide protection like our bones. They can provide transportation like our blood. Both blood and bones are connective tissue. If you think about it, there probably aren't more different tissues in your body than bones and blood. But both of those are connective tissues, and so it gives you some idea of the differences in the structure and the differences of the function. As their names indicate, they're connective tissues, so they're going to bind, they're going to hold things together, protection, they provide energy, they provide all sorts of different things. They're very, very diverse in their function. So again, we've written that on the board, but we can do this way as well. Epithelial tissues cover, they align our surfaces inside and out, they form our glands. Connective tissues protect, support, bind, store energy, transport, provide immunity, and many more things. And like I said, we know muscle tissues contract, and it comes in 3 flavors: cardiac, smooth, and skeletal. And nervous tissue is specialized for communication, it produces action potentials. Here we have it in the words. Here we have it in the pretty picture. Stop it. There we go. Again, showing some examples of all the different tissues and where we would find them. But the last little bit that I want to talk about for today is this part right here. As we talked about, we're moving up our hierarchy. And when we're moving up our hierarchy, tissues are made of cells. And so if we're going to make a tissue, we need to be able to put cells together. And if we're going to put cells together, we do that using cell junctions. And there are 3 main types of cell junctions, and we see all 3 types here. There are tight junctions, which are also called occluding junctions, desmosomes, and gap junctions. What I want to do to finish up today, and we're still okay on time, is I want to talk about these 3 types, identify the components of these junctions, and identify what their function is. And of course, once we do all that, we could kind of come up with some ideas of where you might find them. Let's start first with our tight or our occluding junction. Notice, and again, here we see the picture, and here we see what it looks like in a 3-dimensional cell. When I look at these tight or occluding junctions, they always kind of remind me of my grandmother's quilting. My grandmother's part of this quilting club where they get together, they sit on the porch all surrounding this big, huge quilt, and her friends do all these elaborate quilting stitches. And what a quilt is basically 2 pieces of cloth with some matting under, or in between, so that as they stitch these elaborate designs, it gets poofy in between, and it's beautiful and it's elaborate, and it covers large surfaces. And that's what we see here. The other way to think of these tight or occluding junctions is to think in terms of a zipper. A zipper has 2 sets of keys on the 2 sides. And when you pull up the zipper, what happens is those keys interlock with each other and hold them together. And so the 2 sides of your jacket are held together by those teeth, those keys of the zipper. And that's kind of what this tight junction is. This tight or occluding, again, both are appropriate terms, are these big, long zippers where we have 2 transmembrane proteins, 1 on each membrane, that bind to each other, interlock with each other, and hold the entire surfaces of the cells together. Now, obviously, this is going to provide some structure. This is going to help to give shape to our cells. But the other thing it does, and we can cheat by kind of drawing 2 cells here side by side. So here are 2 cells side by side. This one has an transmembrane protein. This one has a transmembrane protein, and they interlock with each other. And they go long distances over long surfaces of these cells. And I'm just going to draw 4 of them, but you get the idea. And while they provide structure and support to our cell, the other thing they do is tightly, hence the name, hold these 2 cells together. The reason that is important is because here outside the cell are things like water and things that are dissolved in water. And here inside the tissue is water and other things that are stored in water. And if we have a tissue making up an organ, do we necessarily want water to be able to easily pass between the 2 cells and get inside or outside of the cell? No, if these tissues are supposed to provide protection for us, for instance, we want to limit the movement of water, right? How many people have ever washed a dish before? Nobody. Wow, you've all been home. All have maids, take care of it. All right, at least a couple people have. Excellent. Maybe some of you have watched it on TV. It's super exciting. What happens is you take this shriveled up sponge, and the first thing you do is dip it in water. And when you put this shriveled up sponge into water, what happens to the sponge?
Expands.
It expands. It absorbs all that water and expands and gets all nice, big, and poofy, right? How many people here have taken a bath before? Again, none of you. Wow, I do not want to come over and smell your guys' house.
Showers only.
Showers only. Okay. Even as a little baby, you never took a bath? Have you seen
Nope,
someone take
never.
a bath? How about gone into a swimming pool? Anybody gone into a swimming pool before? Is that what happened to you? You went into the swimming pool tiny, and then as soon as you got in that swimming pool, you sucked up all this water and blew up like a balloon? Did that happen to
Well,
anybody?
we absorbed water, but we didn't blow up like a balloon.
Right. You may absorb a little bit of water, but absolutely you do not blow up like a balloon. And the reason for that is things like our skin has a very tight barrier in between the cells using these tight junctions to limit water getting in and water getting out. Right? Think of it the other way around. If you get a severe burn over large portions of your body, right, the two things that they do to severe burn injured people are they keep them in a sterile environment to protect them from pathogens. But the other thing they do is they put them in a humid environment. Because if you've lost large surfaces of your skin, you're going to evaporate water away very quickly. So that skin and those tight junctions in those skin provide important barrier to limit the movement of water in and out. Right? Your digestive system, we want to limit what gets in and out. Your urinary system, right? You produce urine, it goes into your bladder and sits into your bladder until you get to a socially appropriate location. And you don't want to be absorbing all those wastes. So all of these are places where we want to have these tight or occluding barriers because they produce a waterproof seal. So like the skin, our gastrointestinal tract, our bladder, these are all examples of places where we want to produce that tight occluding junction, that waterproof barrier. All right. These are different from our desmosomes. Let's draw our two cells again because I like that. I'm going to do this. Cell 1, cell 2. Desmosomes, if you notice, are much more elaborate in their structure. Notice first there is a peripheral membrane protein. Remember, peripheral membrane proteins are proteins that are not attached to the plasma membrane, but next to them. And notice those peripheral membrane proteins are called plaques. We then have some transmembrane proteins that attach to the plaque, go through both layers, and then have a bulbous structure on the outer surface, both layers of our plasma membrane. So these are transmembrane proteins. There are lots of examples of transmembrane proteins, but the most common transmembrane protein, as we see down here, are called cadherins. Notice there's a lot in a name, adhere. What does it mean to adhere? What does adhere mean?
To
Come on.
stick?
Yeah, to stick.
Together.
They stick. Adhere means to stick to. And these are cadherins because when Ca, calcium, is present, these things become sticky. And isn't there always calcium outside the cell? Yeah, there's always calcium outside the cell, so these proteins are always sticky. In fact, they're glycoproteins. There's a little bit of sugar on them that helps to make them sticky. And so these cadherins stick to each other, interlocking with each other, as we can see here, forming a very, very tight boundary. And notice also, we also have our part of our cytoskeleton, our intermediate filaments. These intermediate filaments, remember we talked about, are the I-beams that help to give structure to a cell, give shape to the cell. They also attach and embed into that plaque. And even though the picture doesn't show it, remember I told you some of these intermediate filaments can actually move from one cell to the next cell. Notice we've got a whole bunch of hunk and junk here holding this cell together, or this cell to that cell, forming a very, very tight attachment. This is important, right? Think of it this way. When you have if you are a welder and you have a big, huge, massive sheet of metal, and you want to attach it to another big, huge, massive sheet of metal, do you attach the entire surface of that entire piece of metal to the second piece of metal? Is that typically what our welders do?
No, just the corners, the side.
True. Or
side.
corners, or 1 or 2 spots. They may do a half dozen spots, 2, 3, 4 spots where they hold it together, what they call a spot weld. A spot weld is one single location where there is a very tight bond, and that one tight location with several others to help can provide massive structure and massive support. And that's what we have here. These desmosomes are basically our spot welds, holding cells together so they resist those shearing and tearing forces. These are tissues that get a lot of use and abuse, right? Like our skin, right? Like any good older sibling, I, like myself, and I'm sure any of you that are older siblings, at one point or another, you gave your younger siblings, right, what is now incredibly inappropriately named an Indian burn, right? How do you do that?
You twist your hands opposite directions.
There you go. You twist their hands in opposite directions on the skin, and it gives them a red burning sensation to them. Now, it hurts them. They cry to mom and dad, but does their skin come peeling off when you do that?
No.
Nope.
No. Why? Because our skin resists those shearing forces, right? One of the things that happen as we age is that these desmosomes break down. While your younger sibling, you can do that to them and not peel off their skin. What would happen if you did that to grandma?
It rips the skin.
Yeah, you might tear grandma's skin if you do that, because her skin has lost some of those desmosomes. It's lost some of that structural attachment. So don't hurt grandma. All right. So we have those tight junctions, right? Another place where we need this is in the heart. The heart is producing massive amounts of pressure to pump our blood throughout our body. And to hold those cells together while they're producing that massive amount of stress, these cells are interlocked with these spot welds, with these desmosomes that hold the cells together. All right, so we have a pretty picture. The plaques, again, are peripheral membrane proteins. We have the integral transmembrane proteins like the cadherin. We have those intermediate filaments that, unlike the picture, can actually cross from one cell to the next. And again, they produce this very tight, resisting spot weld that holds the cells hard together, physically tight together, to maintain the shape and to maintain stress, resistance to that tearing and shearing forces.
Now, is this considered one of the strongest out of the cell junction?
Yes. Of our 3 types of cell junctions, the desmosomes are absolutely the strongest. Yep, absolutely. Especially when you consider it with this one. This third one is a gap junction. Oops, gotta clear my picture. Notice gap junctions are made up again of proteins. In this case, the proteins are called connexons. And as you can see, 6 connexons line up in what is a circle in the plasma membrane. They're a transmembrane protein lining up in a circle on one cell. And notice they line up on a circle in the other cell as well. And what these actually do, notice they're not providing necessarily a lot of integrity. What they're actually doing is providing a passageway. This is a passageway where one cell is able to rapidly communicate with the other. Right? These gap junctions are actually really fluid-filled tunnels. That connect the cells. Notice that's not going to provide a lot of structure and a lot of integrity, but what it will do is allow the 2 cells to communicate with each other. So that what happens in one cell can affect and influence the other cell. So if we go ahead and draw our two cells again, there's one cell, there's another cell. Let's totally cheat and poke a hole. And poking a hole will then allow us to form my connexons. And I'm just going to connect them as one thing. It basically forms a hollow tube that connects the two together, and again is going to allow substances to rapidly move from one cell to another. So if there is a massive increase in sodium in this cell, that sodium can rush to the next cell. Basically, what this allows us to do is to hardwire these cells together. These two cells are connected by fluid-filled tunnels, and so they're going to work together as one, because whatever happens to one cell is going to happen to the other. So in this case, it's not about structure, it's not about shape. In this case, it's about communication. These fluid-filled tunnels allow the two cells to communicate, and it allows them to work together as one. This is really, really important for, like, for instance, again, our heart. Our heart, you produce that, as we were talking about a couple, well, it had to be this last week. As we were talking last week, your heart has those cells that produce the action potentials, that SA node that produces that action potential that spreads it through the heart. And it's able to spread to every single cell of the heart because all the cells of the heart are connected by these gap junctions. So when you produce one action potential, it spreads to all the others, and your heart works together as a single function doing that single pumping action. It can be used in the nervous system as well, but not as much as you would think. And we'll talk about why that's so important when we get to the nervous system as well. All right.
Question.
Yes.
You mentioned that the desmosomes, it's also the heart. And
Yep.
you're saying that the gap junction, it's also the heart too. So is
Well,
the heart...
and isn't, wait, hold on. And isn't the heart also filled with a lot of fluid
Right.
that you don't want to leak out?
Oh, okay, okay.
So wouldn't you think the heart would be a good place for there to be tight junctions as well?
Yeah.
Absolutely. Now, is every single tissue type going to have all three of these? No.
No.
Does cardiac muscle have all three of these? Yes. But not every tissue is. But most tissues will have one or more of these, right? Skin has two of them, the desmosomes and the tight junctions, right? We don't need communication between the skin cells so much, so it only has two of these. Others may only have one. There may be some tissues that have none. But in our tissues, typically most tissues, and again, the keyword is most is not all, but most tissues will have one or more of these junction types. But you're absolutely right. Cardiac muscle has all three. All right. Great questions. Any others? And in fact, I think it's the only tissue that has all three. I can't think of another one off the top of my head.
What
right.
about the brain? What kind of tissue is that?
Well, again, nervous tissue is what we find in the brain. There will be some gap junctions in there, but not as many. Like I said, not as many as you would think. All right, but it's not going to have the other two. All right. With that, we are done with the gap junctions, and so what we have left to talk about is to start talking about the tissues. But that is the point that I'm going to leave for the next class. I wanted to get through the rest of the cell stuff, and this kind of falls under cell stuff because it's talking about how we hold cells together. But now we know how to hold cells together, we can talk about our tissues. And so that is all I wanted to do for today. All right, so where we'll pick up tomorrow is with our epithelial tissues and connective tissues. Again, I want to strongly encourage you to print those handouts out so you can be looking at them while you're looking at the slides that we're looking at on the screen. Yes, you can bring them up on your computer screen, but then you can't see the stuff for the pictures that I'm posting as well. It'll be better if you can look at them separately. I guess if you really have to, you could bring them up on a smart device or an iPad or something. You could have the handout on that and be looking at the pictures on the computer or however it is. But I would encourage you to strongly to print them out. You're gonna need to print them out anyway to do the assignment. So you might as well do it now so that you have it tomorrow so that it'll be easier to understand as we go through the material.
And you did mention that all of them are under the modules, correct? All the papers we need to print out.
So
they're all on the
again, you're right. I mentioned I was going to show you that. So let me do that. Did I close it? Hold on, I closed it, so give me a second. So again, under modules, under your section, this is where you find all the assignments that you need to submit. So this is where you'll submit all your assignments. This is where you find all the lecture handouts. This is where you find all the lab simulations you're going to be doing. This is where you find all the answer keys, the lecture images I'm posting. But what I'm talking about right here are the lab handouts. So this one here, the epithelial tissue handout, when you click on this and you look at it, the very first tissue we're going to look at is going to be a slice of the kidney. And the very first question I'm going to ask you when I show you that picture is I'm going to ask you to identify the specimen. And what do you think the specimen you're going to be looking at is?
Kidney?
Kidney, excellent. The very next thing that I'm going to show you, I'm going to say identify this structure. And guess what the structure is going to be? Anyone? You guys can't see this, right?
The renal corpuscle. Corpuscle.
Corpuscle. And again, so here's the two things I would say with that. Remember, I don't care how you pronounce these things as long as you spell them correctly. All I care about in this class is spelling. I am not a good speller. So one of the things that I did as I was learning all these things is I said them the way they looked because that helped me to spell them correctly. My biggest challenge when I first started teaching was relearning how to say all these things the right way because I'd been saying them wrong in my head all these years so that I would be able to spell them correctly. So I don't care how you say them as long as you spell them correctly. All right? But the point is you've exactly got it. As I mentioned on this study guide for the tissues, not only for simple squamous epithelial tissues are these basically the 7 things you need to know, but they also, in parentheses, have the key word I will ask you for on the exam. So that you know if I ask you for a specimen, what I'm looking for is the kidney. If I ask you for a structure, what I'm looking for is a renal corpuscle. That also means that if I ask you for a space, you know nucleus is never going to be the right answer, right? Because the nucleus is not a space. So again, being the sophisticated student you are, again, you can use my precision to your advantage. Again, some of you already have thought about these things this way. When you took the SATs, like in high school, anybody take the SATs? Anyone ever take one of those like Kaplan classes or something like that to help you with those things? When you take those classes, do they teach you the information you need to know? Maybe a little bit, but a lot of what they talk to you about is how to take a test, a standardized test, right? Like one of the things they tell you is if because it's a standardized test, the format has to be very straightforward. So they always start with the easy questions and then they get harder. So if you're looking at the very first question on the exam and there's 2 possible answers and one of them's obvious and one of them's less obvious, guess the obvious one because it's an easy question. If it's the last question and you have 2 possible answers, one that's obvious and one that's not obvious, take the not obvious answer. Because again, if it's a hard question, that means most people got it wrong. And if most people are getting it wrong, it's not the obvious answer, right? It's about taking the test, knowing how to take the test. This helps you to take the test. If I ask you to identify a tissue, you know kidney is never going to be the right answer. So if I ask you for a tissue, then don't write kidney because you know that's never going to be the right answer. Right? So again, these are the precise vocabulary I will use. And so you're going to want to have these printed out. You're going to have to do the drawings anyway. So you might as well print them out now so that you can have them tomorrow so we can work on them tomorrow. All right? Any questions on any of that?
You did say that we had 2 different sets of papers that we need to print out. So this is one, right? What's the other one that we need to print out? Do you know?
Yes, I do know. So, as I mentioned, you are responsible for epithelial tissues and also connective tissues. two tissues are the two types that are going to be on the exam. Remember, there's 4 tissue types, but muscle tissue we're going to talk about when we get to the muscular system. Nervous tissue we're going to talk about when we get to the nervous system. So for this exam, there's 2 main types of tissues you're going to be responsible for: epithelial tissues and connective tissues. So these are your handouts that are an assignment. These are also your study guide, right? This is the integumentary study guide, the anatomy you're going to be responsible for on the exam. So all of, and histologically for that as well. So for this week, these 3 things are basically your study guide for this week, for this half of the exam. All right?
Okay.
Any other questions? All right, guys, have a fun day, and I will see you tomorrow morning bright and early at 8 a.m.
Thank you. Bye.
All right, take care. No, wait,
Okay.
one question.
Nope.
So yes, actually, you're not the first person who emailed me. Someone else emailed me that they were having problems. Today was the mitosis one, or no, today was the membrane transport one. So remember, you have to complete it. So some of it is trial and error. What I would remind you is if you really get stuck, to go actually and read the information that they provide, because usually they have the right answers in there. But I know sometimes it's a little bit awkward, and if you get stuck, you could lose some points. Once you think you're going to drop below that 80% threshold, then I would encourage you at that point to do exactly what you did. Stop and restart. Or the other thing you can do is, if you get stuck at a part, try to figure it out, try to work your way through. And as soon as you figure out how to get past that part where you're stuck, because I like the Labster a lot, but there are a couple times where it's not as intuitive. And it has you, like, for instance, with some of them, you really have to move around the room a lot. And sometimes it's something as simple as you have to put the pipette holder away before you can pick up the slide. So, I mean, there are some parts that are a teeny bit tricky that way, although you don't usually lose points for that type of stuff. But it can be tricky going through it. So if you get stuck, go through it, try to finish it, not worrying about the points, but at least doing all the steps, and then do it again, and you can do it correctly the second time. If you truly get stuck, also remember, down in the bottom right is that thing where you can contact the at least the bot, the Labster help bot, and they can also kind of provide some advice and help things that way as well. It is a little trial and error of learning, and unfortunately, that is kind of what science is like. So if you really get stuck, then at least try to finish it so that you know what to do, and then it'll be much easier the second time you go through it. Okay? Excellent. All right. Any other questions? All right. Well, then that is, like I said, that is all I have for today. So again, you guys have a good day, and I will see you tomorrow morning. And I'm going to go ahead and stop the recording now.